Interphase is when the cell prepares for division. It's divided into three stages, G1, S, and G2. In G1, the cell grows and prepares for DNA replication by synthesizing the necessary organelles and proteins. After passing the G1 checkpoint, in the S phase, the DNA replication occurs. Each chromosome now has two identical sister chromatids. And then lastly, in G2, the cell checks for errors in DNA replication and goes through a final period of growth and preparation before its final meiotic division.
Similar to mitosis, the process of meiosis consists of prophase 1, metaphase 1, anaphase 1, and telophase 1. During prophase, the chromosomes condense. Homologous chromosomes pair up. Crossing over occurs where genetic material is exchanged between homologous chromosomes. In metaphase 1, the pairs line up at the metaphase plate and are held in place by spindle fibers attached to the centromere. Independent assortment happens here. In anaphase 1, the proteins responsible for sister chromatid cohesion are broken down along the arms, and homologs separate, and the kinetochore microtubules guide them to the opposite poles of the cell. In telophase 1, the chromosomes reach the opposite poles and the spindle fibers break down.
At the end of meiosis 1, each daughter cell contains only one set of chromosomes, each chromosome being of two sister chromatids.
Following meiosis 1 is meiosis 2, in which the second meiotic division occurs. This process consists of prophase 2, metaphase 2, anaphase 2, and telophase 2. The phases of meiosis 1 are repeated and 4 genetically diverse haploid cells from the two daughter cells are produced. The 4 haploid daughter cells each contain a single set of chromosomes.
During meiosis, genetic material is shuffled and recombined through the process of crossing over and individual assortment. This creates genetic diversity among the resulting gametes, which is important for sexual reproduction. The 4 haploid cells produced by meiosis are gametes that can be used for fertilization. When two gametes fuse during fertilization, they form a diploid zygote that contains 2 sets of chromosomes, one from each parent. The combination of genetic material from 2 different parents increases diversity even further and contributes to the variability of offspring.
Meiosis matters because it creates genetic diversity. Meiosis allows for new combinations of genes, which can lead to new traits/characteristics that may be advantageous and become more prevalent, leading to evolutionary changes and new species. Therefore, it is needed for survival and evolution of populations, as it allows them to adapt to changing environments. Meiosis is also important for sexual reproduction, as it is responsible for the formation of gametes.
Meiosis creates genetically unique offspring with the processes of individual assortment, crossing over, and random fertilization. During meiosis, homologous pairs of chromosomes line up randomly along the metaphase plate. When the cell divides, each pair sorts its homologs independently of the other pairs. So each chromosome has an equal chance of being sorted into either of the 2 resulting cells, and each cell will have a unique combination of chromosomes, therefore creating genetic diversity. Crossing over is when homologous chromosomes exchange segments of genetic material, further increasing genetic diversity. Random fertilization refers to the fact that during fertilization, a sperm randomly combines with an egg, resulting in a zygote with a unique combination of genes. The probability of any one sperm fertilizing any one egg is also random, resulting in even more genetic diversity.